Here it is.
Good afternoon, everybody. While we're waiting for folks to come in, please, it looks like everybody has their microphone muted and their cameras off. That's fantastic. Why don't you go ahead and introduce yourselves in the chat so we know who's here. People will keep on coming in, I'm sure, but we'll go ahead and get started with the introductions. So welcome. Thanks for joining us today in the Ohio River Basin Alliance discussion series. Our topic for today is student voices in Ohio River Basin research, and we'll be hearing from three students in the basin. On behalf of the Knowledge and Education for Informed Decisions Working Group Webinar Committee, I want to thank Craig Butler, who's Executive Director of Muskingum Watershed Conservancy District and currently serving as Chair of the ORBA Steering Committee. We also want to thank our sponsors, Orsenco and the Foundation for Ohio River Education. And I want to introduce the members of the webinar working group. Jen Bowman, who's at Ohio University Voinovich School, Chris Lorentz at Thomas More University, Nick Callahan, who's with Orsenco, Ohio River Valley Water Sanitation Commission, Heather Mayfield with Kenton County Conservation District, Heather Sprouse with West Virginia Rivers Coalition, and myself, Chuck Somerville. I am Director of the Ohio River Basin Consortium for Research and Education and Professor Emeritus of Biological Sciences at Marshall University. As I mentioned, we've got three speakers today. I'll introduce them as they come up. We have a graduate student from the University of Pittsburgh and two undergraduate students from Thomas More University that'll speak today. So if you have questions that come up during the presentation, please type them in the chat. I will read them to the presenters at the end of the presentation if we have time, and if we go on to the next presentation, then we'll get to questions at the end of all three talks. We'll have about 15 minutes for presentations today, and then about 5 minutes for Q&A. So without further delay, we'll go ahead and get started with our first speaker from University of Pittsburgh. Her name is Gabriela Zuccolotto. Gabby's a second-year PhD student in the Department of Geology and Environmental Science at Pitt. She's interested in hydrology, remote sensing, and her current research utilizes long-term satellite records and machine learning to study harmful algal bloom dynamics in the Ohio River Basin. She's a former middle and high school educator, and she's passionate about science, communication, and outreach. Gabby, thanks for joining us today, and take it away.
Thank you so much. Can we retry the screen share one more time because I'm not seeing PowerPoint listed in one of my applications? Thanks, Nick. Okay, still not showing up, so I'm just going to share my regular screen and hopefully it doesn't turn green. Not sure why that happened, Nick. Okay, hopefully everyone's able to see my presentation. Thank you. Awesome. Thank you so much for the introduction. As was mentioned, my name is Gabby Zuccolotto. I'm a second-year PhD student in Dr. John Gardner's lab at the University of Pittsburgh, and I'm really excited to share some of the research I've been doing for my dissertation, looking at our ability to use satellite remote sensing to study algal bloom trends in the Ohio River Basin. So the Ohio River Basin has experienced several large algal bloom events in recent years. In August of 2015, there was a large algal bloom event that lasted several months and stretched 2/3 of the Ohio River's total length, which is 981 miles. This event was about 700 miles long. And you can see in these photos on the right how severe that 2015 bloom event was. Stretched multiple states, led to multiple recreational advisories, and at the time it was called an unprecedented bloom event. Then in 2019, a second bloom event also occurred in the main stem, Ohio, not quite as long, about 300 miles long, stretching from Ashland to Louisville. But both of these were large, unprecedented events, and there's been a lot of work done by different organizations trying to figure out what led to these bloom conditions and how we can best manage our rivers to prevent them from occurring. The reason we're concerned about harmful algal blooms and algal blooms in general is that some species of algae can produce neurotoxins, which can be harmful to human health, harmful to your pets, aquatic life. They can contaminate your drinking water, and they can also lead to recreational advisories. And anecdotally, we're seeing that river algal blooms seem to be getting bigger, more frequent, more severe, and this seems to be a global phenomenon. The problem is that river algal blooms are really understudied, and we have little information regarding their frequency, their magnitude, their drivers, and the conditions that are leading to these events occurring. So these two plots were made by an undergrad at Pitt, Rob Murphy, who did some archival work for me, and he was looking at in what sort of environments is most of our research done on algal blooms being conducted in. And what we found is that a lot of our knowledge from algal blooms comes from lake and coastal systems, and there's very little research that's been done in terms of understanding these blooms in streams and rivers. We can also see that geographically, we have a lot of spatial bias. And like I said, even though anecdotally, we know that these river blooms are happening all around the world, we do see concentrated areas where a lot of the research is being done. So our overarching goal of this project is to really improve upon that knowledge and understanding of river algal blooms. So how do we monitor these algal blooms? One way we can do so is using chlorophyll A. Chlorophyll A is a pigment that's found in plants, green algae, any organism that undergoes photosynthesis. And we can use concentrations of chlorophyll A to understand algal biomass, or essentially how much algae is in the water. So you can see in these photos here on the bottom, this picture, you can see the algal cells and what that chlorophyll actually looks like. And most of the time we're getting these chlorophyll concentrations from grab samples, so actual samples of river water that are taken out and processed in a lab to understand how much chlorophyll was in the water at that time. But we'll see when we look at this map on the right that chlorophyll A grab sample data is fairly limited in the Ohio River Basin relative to other major river basins in the east. So we don't have a ton of grab sample data that's available to us. So what can we do about that? That is where remote sensing comes in. Chlorophyll A is used commonly in remote sensing studies because it's detectable with satellites. That's because chlorophyll A has a distinct spectral signature that we're able to detect with satellites in space. So we see that chlorophyll A absorbs electromagnetic radiation really strongly in the violet and blue visible wavelengths, as well as the orange to red wavelengths. Using this to our advantage, our goal was to, one, build a chlorophyll A database using NASA's Landsat mission calibrated to the field data that we have available to us, and then using that to generate observations averaged over every river reach in rivers that are larger than 50 meters wide. The reason that we're focusing on large rivers, rivers that are greater than 50 meters wide, is because of Landsat spatial resolution, which is 30 meters. So we need rivers that are actually visible to the satellite. How we actually are producing that database, and I'll talk more about this in a moment, is coincident measurements of our in-river chlorophyll A data and pairing that with satellite passes that are taking place within 48 hours, either before or after of when that in situ measurement is being taken. Our second objective is then to use that database that we produce to understand how chlorophyll A concentrations have changed across time and space in the basin. I do want to mention one caveat before I continue, which is that all of these results are preliminary. This is an ongoing project, part of my dissertation work. So both our model and some of the trend analysis that I'm going to show does need some improvement, but I thought it was good enough to share with everybody. And one major thing to keep in mind is that our model is currently running into some challenges predicting chlorophyll A concentrations for really extreme events. So keep that in mind, like I said, as we look through some of the trend analysis. So these are some of our model results. This figure on the left shows the location as well as the amount of data that was available for those matchup pairs. So these are places where we have an in situ measurement matched to a satellite overpass. So this is what we used to build our model. We ended up with almost 80,000 matches across about 7,000 sites from 5 major river basins in the East. We should note that we did filter this data to anything that's below 200 micrograms per liter in concentration. And again, that is trying to filter out some of those more extreme events. And now we're working to improve that and be able to filter some of that data back in. So that's what we started with. What we ended with is shown on the right. These are our model results. So this figure in the bottom shows what we ended up for our correlation between our measured chlorophyll A concentrations and then what our model was able to predict. We have an R-squared value of 0.79, which means it's a pretty good correlation. It has pretty good prediction capability. And then this table up at the top is an example of what our tabular database actually looks like. So we have some sort of reach ID that's linkable with the National Hydrography data set, if any of you are familiar with that data set. So we'll have our reach ID, the date of that satellite pass, and then the predicted chlorophyll A concentration. What's great about this database is that because we're able to calibrate that model and then apply that model to all of the data that's available across Landsat 5, 7, and 8, Landsat 5 started in 1984. So at this point, we have almost 40 years of chlorophyll data at the reach scale level, which is really great. So here are some of the preliminary results for more of the analysis part of that database. So this plot here shows the Ohio River Basin. You can see where Pittsburgh, Cincinnati, and Louisville are located at. And then all of these lines, the colored lines, represent our Landsat visible rivers split up into those individual reaches. So we have 5,257 individual reaches within the Ohio River Basin, and altogether we have almost 3 million reach scale chlorophyll A measurements across that 40-year record or across this large database. The coloration on this map represents the counts for each of those reaches that are available, so our number of available chlorophyll measurements. And what I want to point out in this is that because we're working with satellite data, we do see some spatial and temporal skewness when we look at that. So you can see that certain reaches darker in color have more data available than the reaches that are lighter in color. And that's just something to keep in mind as we move through the analysis. So both of these plots represent essentially sanity checks for what we know about how river blooms function. And about how algal blooms in general function. So this plot on the left that's colored right now shows our temporal trends. This is all of the data aggregated by month for the entire river basin. And what we can see is that during the warmer months, we're getting an increase in where our peak or our most dense chlorophyll concentrations are. And then we sort of see that start to degrade or go back down in the colder winter months as that algae is less productive. So again, a good sanity check for us that the database that we've created makes sense with what we know about how algal blooms work in the environment. This plot on the right then shows our mean chlorophyll A concentrations for every reach across that entire record. And this is more of a spatial view of this. And we can see that certain subbasins in the Ohio River Basin are more chlorophyll productive, you could say. So I have two of those pointed out. This larger circle is the Wabash Basin. And this is a basin that we know has a lot of agricultural activity. It has a lot of nitrogen and phosphorus loading. So we know that there are active blooms in this area. The smaller circle at the top, where you see this darker green part right at the upper part of the Allegheny, that's the Allegheny Reservoir. And that's another place that we know in recent years they've had an uptick in bloom activity and that there's been bloom activity happening somewhat frequently. So we would expect to see higher concentrations there. We're also interested in looking at the severity of these blooms across time and space. So this stacked bar plot on the left, you can see our years going across our x-axis, and each one of these represents a bloom season. So this is the data from May to November for each one of these years. The coloration then on these bars represents how many of the observations across the whole river basin during that bloom season were either severe or non-severe. And we're using a cutoff or a threshold of 30 micrograms per liter or above to indicate a severe bloom condition. The reason that this plot is interesting is because, again, it highlights one of the challenges you could say associated with using this database, in that because we're using remote sensing data from 3 different satellites, we see the amount of available data change. So we do see an increase whenever, 1999, when that was the introduction of Landsat 7. So we see an increase in the amount of available data. So that's important to keep in mind when we're looking at these percentage of severe occurrences. Looking at severity from a spatial perspective, we again see some really interesting trends. So in this case, our color scale represents the percentage of observations for each reach that indicated a severe condition. So in the Wabash Basin that I pointed out earlier, you can see, particularly in some of the downstream parts, out of all of the observations available for that subbasin, essentially 50% of them are indicating that those reaches are above 30 micrograms per liter. I think that's everything for that. And these are results of some trend analysis that I've done. For those of you interested in statistics, these are Mann-Kendall trend tests. So I approached this in 2 different ways. I first looked at how the mean is changing for each of these river basins across time. And then I also looked at how the maximum is changing across time. So this figure on the left shows our mean bloom season. So again, just from May to November, how, again, those means concentrations changed across that 40-year record. And what we found is that about 44% of reaches in the Ohio River Basin, when we're looking at the mean, are decreasing over time. These are really noticeable, particularly in the main stem of the Ohio and the lower Cumberland River in the southern part of the basin. Not many of the reaches are increasing when we look at the mean, and then there are about a little over half of the reaches that aren't trending one way or another. So whatever their chlorophyll concentrations are, they're staying fairly stable across the record. When we're looking at our maximum trends, though, we see something different and very interesting, in my opinion, which in this case, when we look at how the maximums are changing across the bloom season, only about 10% of the reaches are decreasing, and we see an uptick in the number of reaches that are actually experiencing an increasing maximum. So this could be indicative that the chlorophyll concentrations are getting more severe over time. What's really interesting is that when I looked through where some of these increases were actually occurring, many of these are isolated to lakes and reservoirs within the basin. So this one in the north is Huntington Lake. We have the Upper Allegheny Reservoir in the northeast part of the basin, and then Lake Cumberland in the southern part. And then we see again that we have a lot of reaches that aren't trending one way or another in terms of their maximum. So what's next, along with, like I said, trying to make some adjustments on the model to make sure that we're predicting high chlorophyll concentrations well, and we're capturing those really big bloom events in 2015 and 2019, we also want to test our conceptual model. Much of our knowledge from lakes and reservoirs indicates that blooms tend to happen as a response to increasing nutrient runoff, particularly nitrogen and phosphorus. What we're seeing in the Ohio, though, when we look at this photo on the right, these 6 plots, is that we do see seasonal variation, but in general, a lot of our total nitrogen and total phosphorus in the main stem Ohio is actually declining over time, which means then that there could be some other processes at play here that could be leading to these large bloom events. Some preliminary work that has come out of Orsenko and USGS, some work here being done at Pitt, all sort of indicate the same thing, which is we think that the relationship between high flows and low flows, high flows occurring in the fall, coming after low flows in the summer, that could be leading to high summer flows followed by low fall flows could be leading to an influx of nutrients that are then essentially sitting stagnant in a low flow condition, and that could be leading to bloom proliferation. So we're testing out how our river management, how our flow conditions, and how our nitrogen inflow is possibly affecting the conditions that are leading to blooms. That's everything I have. Thank you so much. I'll take any questions, and my contact information is there if you have any questions that you'd like to talk one-on-one about.
Thanks, Gabby. I think that was a great talk. We've been interested in blooms in the Ohio for several years now, and one of the things that we ran into with satellite imagery was a high number of days where there's cloud cover that makes it difficult to get good images. Have you experienced any of that? And kind of a corollary to that is if you're working on measuring a bloom that's either developing or already in existence, does that cloud cover issue get in the way?
Yeah. So cloud cover is something that has been difficult to deal with. The way our model is set up is that we do remove any cloud cover in each image as well as cloud shadow. So we're trying to work around that. Unfortunately, if the entire image is cloudy, there's not much that we can do, but it is something we do have worked into the algorithm. And we're hoping that, if there's some available data that at least long-term trends, we should still be able to look at.
Yeah, thanks very much. Again, great talk. I'll wait to see if any other questions come up for you in the chat. Thanks. Meanwhile, we're gonna move on to speaker number 2. And speaker number 2 is Matthew Wimmers from Thomas More University. Matt is a senior at Thomas More University with research interest in native freshwater mussels and fish host ecology. He is, after graduation, hoping to pursue our master's, continuing his work with native mussels. So, Matt, when you're ready, please take it away.
Can everybody hear and see my slides, or hear me and see my slides?
We're hearing you right now, Matt. We're not seeing the slides right now.
You were sharing your email instead of the slides, so I went on ahead and took it back and sent it back to you so you can change that over.
My bad. Can you see the slides now?
Not yet.
While Matt is figuring that out, there's actually a question in the chat for Gabriella.
Yeah, thanks, Nick. Good catch. So the question is from Stacy Burkhardt, and the question is there nitrogen and phosphorus, are those the only causes for blooms, or are there other things that cause the blooms outside of low flow? So
Yeah, I can speak.
Yeah, go ahead.
I can speak to that real quick. So we mainly focus on nitrogen and phosphorus because that, in our understanding of lakes, has been what historically drives these blooms. But it is possible that those low flow conditions that you're getting not only deeper light penetration, but also warmer temperatures, and then also just climatic change driven warmer temperatures could also be contributing to bloom growth. So yeah, outside of nitrogen and phosphorus, we're really concerned about low flow and temperature.
Great, thank you. Okay, Matt, when you're ready, go ahead and take it.
Can you see my slides now?
Yes.
Okay, good. Yeah, like you said, my name's Matt Wimmers. I'm an undergraduate at Thomas More University, and I do a lot of my work at the Center for Ohio River Research and Education, which is the Thomas More University Biology Field Station. And my last project I did there was the reconfirmation and discovery of suitable fish hosts for Lampsilis fasciola, the wavy-rayed lamp mussel. It's a little bit of an introduction. So our native mussels here in North America belong to the family Unionidae. So many of you probably have heard or know about zebra mussels or Asiatic clams. And yes, those are aquatic bivalves, but they're not native to North America. Our native aquatic bivalves are in the family Unionidae, and we have roughly 300 species here in North America, and they're very unique because of this unique life cycle that they have where they require a fish host to reproduce. So like I said, they require this larvae stage to attach to a fish host where they where they kind of spend 2 to 3 weeks gaining nutrients from the fish where they can metamorphosize and grow. And after that 2 to 3 week time span, they'll fall off as juveniles, where then they'll begin to grow and live the rest of their life as benthic organisms. And there's 3 ways that native freshwater mussels get their larvae or Glaucidia onto the fish host. And probably the most common way that we know of, because it's oftentimes depicted in textbooks and like seminars and stuff like this, is their fish-like lures that they possess. So the female, when she becomes gravid or containing the larvae, she will grow out her mantle, and it'll mimic oftentimes a fish-like lure. Sometimes they'll mimic invertebrates as well. And in the bottom right here, you can see this is a picture of the mussel species that I worked with, Lampsilus fasciola. And that's its mantle that she's grown out. And it mimics a small fish or a minnow. And when she's ready to infest the fish with her Glaucidia, she will move her way up from beneath the substrate, and she'll kind of like put this mantle out, and she'll wave it back and forth to draw in a fish. And then when a fish strikes it, she'll rupture her gills, which is where the Glaucidia is housed, or that's where they're kind of maturing during that time until she's ready. And once that fish strikes, she ruptures that, and it kind of shoots the Glaucidian larvae all over the fish to where it attaches to its fins and gills, and that's where the life cycle starts. And there's 2 other ways that mussels can get their larvae onto the fish, and that would be conglutinates, which are Glaucidia-filled packets. It's got a membrane around this packet, and it's got thousands of these larvae inside of it. And these larvae also mimic prey items of the fish that they use as hosts. So they've co-evolved to mimic what their fish host would prey upon. So they can look anything like macroinvertebrate larvae, fish eggs, anything that a fish would want to eat. And then once that fish attacks that, it'll rupture, just like if it was a lure where she ruptures her gills and it finds their way onto the fish. And the third way is just simply broadcast spawners, and that's where when the female is ready to release her Glaucidia, she'll just release them into the water column, and they'll kind of float with the current until they find the fish that they hope would be the host. And fish species varies by mussel species, so not every fish species works for every mussel species, and we do not know every species of fish for every species of mussel. So that's what this project was aimed at, was reconfirming and see if we could identify any new fish hosts for this mussel species. So the methods were, first we went out and electrofish for our fish to use as our study individuals. And once we had our fish, Dr. McGregor from Kentucky Fish and Wildlife was able to go out into the field and collect 2 gravid females of Lampsilus fasciola. And once he found those mussels, Jonathan Frohmeyer, his PhD student, brought them up to our field station, and him and I extracted the Glaucidia. So that was done, seen in this first picture. We use a headlamp, we slightly pry her open, find where the enlarged gills are, where the Glaucidia is housed, and we take a syringe and a needle, and we puncture the gills to release the Glaucidia, and then we flush them out into a dish, which is seen here in the second picture. Once we have our Glaucidia extracted, we then took our fish individuals and we knocked them out, like put them under like how we would get put under if we were having a surgery. But we used 2 liters of water and 0.3 grams of MS-222, which is just a chemical that causes the fish to lose its equilibrium, which is seen in that picture. That's a rock bass that was in the water in that solution for approximately a minute. And once they start to lose their equilibrium and fall on their side, they're pretty well knocked out and they're not going to move too much. And we do that for the safety of the fish, as well as making the process go a lot smoother, because the next step would then be to lay the fish out into a larger dish, and we slightly lift its operculum up to have access to its gills. And we take a pipette with where we took up some Glaucidia, roughly 5 to 10,000 Glaucidia per pipette, and we flush it over the gills. And we do kind of a lot compared to what's used in propagation methods. That is because A, we're trying to see if it really works, because not all the Glaucidia will attach, and B, having too much can oftentimes stress the fish out, and that can cause them to actually shed the Glaucidia. But for a study like this, we try and load as much as we think we can get on there safely. And then once we have our fish inoculated or infested with the Glaucidia, we place them back into a bucket where they can recuperate for 5 to 10 minutes before placing them back into their tanks. And then once they're back in their tanks, we let them sit for a couple of days so they're not stressed out, because stress is a big problem in them shedding the Glaucidia. And after a couple of days of that, we begin siphoning to clean the water back up for them, because water quality is another big issue for them shedding Glaucidia. And then after 5 days, we took the fish back out of their tanks, knocked them back out with the MS222, and then we placed them under a scope. And what we were looking for is what's seen in this image on the left, and that is all those white dots on the gills there are encysted Glaucidia. So after 5 days, if their Glaucidia is encysted, we can confirm them as a potential host. Most of the time, if they're not going to work as a host, they will shed all the Glaucidia within those 5 days. So what we were looking for again were those white dots, and those white dots are actually larvae mussels that are encysted and are gaining nutrients from that fish. So once we identified that, we placed those fish back into their tanks, gave them a few more days, and starting on day 12, the tanks were siphoned every day. The bottom of the tanks were siphoned, and all the contents, whether that be detritus, excess food, waste, or what we were hoping to find, juvenile mussels, were all at the bottom, and we siphoned those through a very fine mesh screen. And then we took the contents of that, placed them under the microscope to hopefully find juveniles. And juveniles were confirmed to be seen if, in this picture on the right, if we saw a visible foot, which is part of their anatomy. That's how they dig and hold themselves in the substrate. So the larvae don't have these, the foot, as well as in the bottom left of this picture, you see these clear dots, and these are the formed abductor muscles. The larvae will have just one big white, but the indication of the two clear spots is the formation of the adductor muscle, as well as the visible foot present on the juveniles. So the results here in this table, we can see that in the first column here, we see our known fishes that are known hosts from literature. And it's primarily the sunfishes, particularly the black basses. So we have smallmouth, spotted bass, and largemouth bass, as well as rock bass and long-ear were known to work. And after the 5 days, the black basses had attachment, but the rock bass and the long-ear did not. And then we recovered juveniles from all of the black basses. And then not known species, or species we were testing, were the green sunfish, a hybrid between a bluegill and a pumpkin seed, and and warmouth. And all of them had encystment on day 5, so we confirmed them as potential hosts. And the only one that we confirmed as a potential host that did not produce viable juveniles was the warmouth. And then here is a table showing the amount of juveniles we recovered off of each individual fish. And again, we see that the 3 black basses produced a lot. They are very robust hosts for this species, which is by no surprise given their lure. Like from the previous slide that I showed you, it's very minnow-like, and bass feed heavily on minnows. So they co-evolved that. So it was no surprise to see them work very well for this mussel species. And then we see that the green sunfish, which was not known, is also a very robust host. And again, this is to no surprise, because green sunfish are a very aggressive sunfish with that slightly larger mouth compared to like a bluegill or a long-ear. And then also, The hybrid sunfish worked as well, but it produced not as many. And some literature suggests that the bluegill does work as a host, but is not a very robust host. And then also the pumpkin seed was not known, but it can be assumed that it is known given that it did work in this hybrid situation, but further testing for that would need to be done. And then, like I said, the warmouth, they had encystment on day 5, but they did not produce any juveniles. So the conclusions were, again, we reconfirmed the black bass species, the largemouth, smallmouth, and spotted bass, confirmed 2 new species not found in literature, green sunfish and the pumpkin seed, that part of the hybrid. And then also we found that warmouth, which was not known to be a potential host, and further testing for those to confirm them as a host will need to be done. And all this is important for propagation efforts for conservation. So facilities like where Dr. McGregor works, they put a lot of work into understanding this fish-host relationship so that they can propagate mussels, oftentimes very rare or endangered species. And they are able, by them understanding what they use as hosts, they're able to propagate these in a hatchery so that they can then go and put them back out into the wild. And I would like to acknowledge Dr. Lorenz and Molly Williams at the Thomas More Biology Field Station for allowing me to conduct this research and using the facilities. I would also like to thank Dr. McGregor from Kentucky Fish and Wildlife for providing the mussels, and Jonathan Frohmeyer for his expertise and assistance. I would also like to thank anybody who helped, including Sarah Kerr and the rest of the 2023 summer interns. And that's all I got. If you guys have any questions for me.
Thanks, Matt. Really interesting talk. There is one question in the chat now. The question is from Joe B, and it's, can bowfin and gar also be hosts? And I'm assuming it's for this specific mussel.
For this specific mussel, what I have found is that they could be. I don't think they've been tested, but I'm going to say probably not, given especially the gar's lifestyle. They're more of, middle of the water column type feeders. Not to say that they won't go on the bottom to eat something, but just given that idea of that coevolution, I would say they could work theoretically, but I don't think anybody's tested it. But I would not be surprised if they also did not work. But I do know that gar do work for some species of mussels, and I have not heard of bowfin working for any mussel species, but that again is something that probably A, hasn't been tested much, and B, kind of just hasn't been tested. So I've not really read anything about bowfin, but certainly interesting to think about and possibly try for a species.
Great. Thanks very much. If there are any other questions, please go ahead and type them into the chat. Kind of quick question. This may be kind of tangential to your work, but in oysters, there's for those juveniles, for the spat to settle, there's specific surface requirements. There has to be an active biofilm on the surface that they settle. Do you know anything about? Has that been studied in mussels in the Ohio River?
I'm not sure specifically on the Ohio River, but there has been a lot of work on like the requirements for the larvae and the juveniles, and they're very sensitive to different pollutions and sediment types. So like if your fish, like the fish that is infected, if it swims somewhere and drops that juvenile where, it's high silt or something, it's probably not going to do very well. And then like in the hatchery setting, like Dr. McGregor has all this stuff figured out to where it's like to a T, like ammonia, water temperature, alkalinity, like all of that stuff has to be to a T to get the results that they need. And then again, that goes into like part of the reason why we could be seeing a decline in our native mussels is they aren't having these requirements that they need in this life stage.
Yeah, that's great. That's the kind of the point I was hoping you'd mention is that as we work on conservation and restoration in the Ohio River Basin, it's not enough just to make sure that the host species are present or that the mussels are present, but there are a lot of other conditions that have to be met. So thanks for that. we'll go ahead and continue to type questions in if you've got them for Matt. We'll move on to our third presenter, Lily Daniel. Lily's also from Thomas More University. She's a junior at Thomas More. She's a dual major in life science with a concentration in sustainability and education studies. She also minors in psychology, environmental science, and biology. So Lily, pretty busy. Thanks for taking time out to be here with us today. After graduation, she's interested in career in conservation, outreach education, and environmental consulting. Thanks, Lily. It's all yours.
Thank you so much. Let's see. Can everybody see my slides okay and hear me okay?
Yes.
Okay. So this previous summer, I was an intern with Boone County Conservation District, and my supervisor was Susan Brown, and together we put together this research study, a comparison of bat acoustic monitoring survey methods within an Ohio River riparian forest. So bats provide important services to an ecosystem, including pollination and seed dispersal. The diversity of bat species present in an area is an important indicator to the health and stability of the ecosystem. The purpose of this study was to gain better understanding of our forest health to make wise land use decisions. We collected this baseline data about species diversity within northern Kentucky and evaluated the impact of unique habitat based on geology to this diversity. So 2 ecosystems were evaluated as a part of this study, the Ohio riparian forest and inland habitat in Boone and Campbell County, Kentucky. The Ohio riparian forest has an open and closed canopy woods and a valuable water source. This forest buffer provides conservation benefits and wildlife habitat. The 3 properties I studied were, so the parent property, at the time it was just called the parent property, but it's now officially called the Ohio River Conservation Corridor at the Northern Kentucky shore. This is a habitat preservation project by the Boone Conservancy. This project is protecting nearly 400 acres of forest, wetland, and riverfront property. I also worked with the Thomas More University Biology Field Station and then St. Anne's Woods and Wetlands, which is connected to the Northern Kentucky University Research and Education Field Station. I am extremely lucky to work with 2 field stations. Field stations provide students with unique research opportunities and hands-on education. The inland habitats include areas defined by glacial deposits, limestone, sinkholes, rock caves, and large canopy forest. Boone Cliffs is especially notable in Boone County for its quaternary glacial conglomerates, with cave-like structures and protected holes for roosting. So at each property, I followed trails to an open area with a nearby water source and snags. For stationary monitoring, the Anabat Swift was hung on a tree for about 5 feet off the ground with a master Python cable lock and left there for a minimum period of 7 days. The walkabout and echo touch meters are handheld devices used to walk around the selected areas and collect live data. The acoustic monitoring devices generate wave files containing echolocation pulses. This data was run through Kaleidoscope Pro analysis software, which is an acoustic analysis program. The program matches echolocation pulses to known structure of species. Spectrograms of the call are generated for manual review. Anabat Insight software was used as an additional confirmation of species identification, and the data threshold was set to a minimum of 5 high-quality pulses and at least 2 call files to notice species. After week-long stationary surveys and mobile acoustic monitoring of echolocation calls, a total of 11 species were detected across 5 sites. And as anticipated, the big brown bat and hoary bat were detected at all locations. It was surprising to me that the Indiana bat was not in all the wetland locations. And keep in mind that the Indiana bat is one of 3 endangered bats in the state of Kentucky. This could also be due to noise disturbance on the Ohio River, and I would suggest further research to be conducted on this. And I also learned while Kaleidoscope is a great tool for automated bat acoustic identification, it lacks some useful features as a call viewer. So the inland sites contained 11 species as compared to 6 or 7 at the riparian sites. The Kaleidoscope software is not necessarily made to detect any of the big-eared bat species, so these are also known as whispering bats. So these 2 species are the Virginia big-eared bat or the Rafinesque big-eared bat. Running these calls through a different software, Anabat Insight, showed 2 important factors for differentiating the calls. The spectrograms are too intense of a call for a whispering bat. The second key point is the shape of the call. So when we look at the calls, the big brown bats have call shapes in the shape of a J, while big-eared bats have small convex humps. Another example showed that within one call, there were 3 different species. For example, we saw a big brown bat, a horay bat, and a tricolored bat, and these could have been social calls or search space calls. Kaleidoscope can only show 1 species per call, and Anabat Insight is really helpful for species confirmation and determining a theory to what is happening in an acoustic. For the future, studying the diversity of bat populations is an indicator used to monitor the health of an ecosystem. Future research opportunities include continued study over time to monitor these changes in the diversity presented in this study. There's also the opportunity to further confirm the diversity by bringing in other monitoring methods like mist netting. And then improvements on the stationary monitoring should be longer time and more sites, because I would love to explore more sites and see what kind of other diversity we can compare. And the Ohio River is an ecosystem vital to the region and encompasses more than just what lives in the river itself, which is really why I wanted to study bats. And then these are all the sources I used. And then these are some of my acknowledgements. All of these people helped me along the way and inspired me to continue my love for bats and just made me want to do so much more research. So thank you. And a special note to Susan Brown for helping me and supporting this project and mentoring me. I really appreciate you. And then this is our contact information. And that's all I have.
Thanks, Lily. I think we talked about, I think you and I talked about your study at the ORBA meeting in the fall. And I'm really intrigued by your hypothesis that sound on the Ohio River might have an impact on bat populations. Have you seen any more about that or thought any more about how that could be studied?
I have not, but it was suggested to me that at some point we try a river route. So we get on a boat and take the walkabout device and connect it, and it's a vehicle across the Ohio River sometime at night. We did do a driving route at one point where we were in the Boone County Conservation van, and we had a speaker attached to the top of the van, and we were collecting live data while driving around. So I think maybe doing it on the river would be really interesting.
Yeah, I want to also let you know that there's an acoustic ecologist at Marshall University, Dr. Ann Axel, A-X-E-L, if you want to kind of reach out to her. She does field work in Madagascar, but we've also had talked about the theory that where rivers run close to highways and other very noisy places, that might have a big impact on lots of the local kind of biota, both around the river and in the river itself. So she is interested in doing some work here in the States. There's a question from Dr. Sluss in the chat. It's what's the cost of the software and equipment?
The equipment is very expensive. I don't know the exact cost, but the Echo Touch meters that just plug into our phones that we can use, those are about $200. And then probably the boxes are like $500 to $600.
Yeah, and there was a message in the chat for Gabriella. She looks like she picked that up. Good. Any other questions for Lily? There's a comment in the chat. This Kaleidoscope Pro software is a $500 per year subscription. Well, I want to thank you for reminding us also that that the quality of the habitat, when we're talking about issues in the Ohio River Basin, we're not just talking about the what's in the water itself, but the upland species and the upland conditions are really important as well. So thanks for keeping that in focus for us. We still have 10 minutes to go. If there are any other questions at this point, I invite you to unmute your microphone, turn on your camera, and ask the question of the student that you're interested in talking to. All right, if there aren't any further questions, students, would you mind also once again sharing your contact information in the chat? Therefore, well, that way, if somebody wants to follow up with you a little bit later on, they can do so. So Gabby just did that. She was thinking ahead. All right, I'll invite Jen Bowman or Chris Lorenz or any other folks from the webinar committee who are in the audience today, if they have any comments or information for the audience, please go ahead and add it now.
Thanks, Chuck. Can you hear me okay?
Yep, you're coming through great.
Just wanted to thank all of our presenters today and let the folks know that are on the call here that this is concluding our last of our 3 webinars that we conducted for the winter webinar series for the Ohio River Basin for 2024. If there are topics or presenters that you would like to get in the queue for our 2025 series, please reach out to me, Chris, or Chuck, and we would be happy to arrange for you to be one of our presenters for next year. Chris?
Yes, I'd like to add my thanks to all the students. Great presentations, very inspiring. And also add that in the fall, in November, we're going to be having an Orba Summit at the University of Pittsburgh, co-hosted by Andy Quinn on the MON project, John Burgess and Megan Lang at University of Pitt. And we love student presentations. So faculty, please let the students know this is a very student-friendly atmosphere for both undergraduates and graduates. And that is our annual summit, which will be in November in the University of Pittsburgh. Looking forward to that. And thanks, Jen, Chuck, and all the KID committee members.
Thanks, Chris. Yeah, that's a great point. We'd love to have students involved in the meeting in the fall. It is a great place for to start your networking as you work through school and go on into your professional careers. So please keep your eye on the Orsenco website and the Orba website for more information. Thanks to everybody. I think that concludes today. There was a question from Dr. Sluss again in the text or in the chat, but Lily made an answer there in the chat. It was about whether or not the study required a rabies vaccination, and that would be the case if the students were physically handling the bats. All right. Thanks for attending once again. Thanks very much to the students for presenting today. Thanks to Chris and Jen and all the members of the webinar committee for the work this year. Very much appreciated. Thanks, everybody.
Thanks. Bye-bye.